Everyone, welcome to the video lecture for Unit 5, Module 11.4. In this one, we're going to discuss protein metabolism. As always, here are your learning objectives. Be sure to reference these when studying for both the lecture quiz and the unit exam. Let's go ahead and get started. So the body can synthesize 100,000 to 140,000 different proteins. They have various functions and structures, yet each protein contains some combination of the same 20 amino acids. Under normal conditions, cellular proteins are continuously recycled in the cytosol. Peptide bonds are broken, and the free amino acids are used in new proteins. If other energy sources are inadequate, mitochondria can generate ATP by breaking down amino acids in the citric acid cycle. Not all amino acids enter the cycle at the same point, however, so the ATP benefits vary. Nonetheless, the average ATP yield per gram is comparable to that of carbohydrate catabolism. The first step in amino acid catabolism is the removal of an amine group, or the NH2. This process requires a coenzyme derivative of vitamin B6, or pyroxidine. The amino group is removed by transamination, or deamination. We consider other aspects of amino acid catabolism in a later section. Transamination attaches the amino group of an amino acid to a keto acid, which resembles an amino acid except that the second carbon binds an oxygen atom rather than amino group. This transfer converts the keto acid into an amino acid that can leave the mitochondrion and enter the cytosol. There it can be used for protein synthesis. In the process, the original amino acid becomes a keto acid that can be broken down in the citric acid cycle. Cells in many different tissues perform transaminations, enabling cells to synthesize many of the amino acids needed for protein synthesis. Cells of the liver, skeletal muscles, heart, lung, kidney, and brain are particularly active in protein synthesis. They carry out very many transaminations. Deamination prepares an amino acid for breakdown in the citric acid cycle. Deamination is the removal of an amino group and a hydrogen atom in a reaction that generates an ammonium ion, or NH4. Ammonium ions are highly toxic, even in low concentrations. Liver cells are the primary sites of deamination. They also have enzymes that remove ammonium ions by synthesizing urea, a fairly harmless water-soluble compound excreted in the urine. The urea cycle is the reaction sequence that produces urea. When glucose supplies are low and lipid reserves are inadequate, liver cells break down internal proteins and absorb additional amino acids from the blood. The amino acids are deaminated, and their carbon chains are broken down to provide ATP. Three factors make protein catabolism an impractical source of quick energy. One is that proteins are more difficult to break apart than are complex carbohydrates or lipids. Two, one of the byproducts, ammonium ions, is toxic to cells. And three, proteins form the most important structural and functional components of any cell. Extensive protein catabolism threatens homeostasis at the cellular and systemic levels. Several inherited metabolic disorders result from an inability to produce specific enzymes involved in amino acid metabolism. For example, individuals with phenylketonuria, or PKU, have a defect in the enzyme phenylalanine hydroxylase. They cannot convert phenylalanine to tyrosine. This reaction is an essential step in the synthesis of norepinephrine, epinephrine, dopamine, and melanin. If PKU is not detected in infancy, phenylalanine builds up and the central nervous system development is inhibited. Severe brain damage can result. The condition is common enough that a warning is printed on the packaging of products that contain phenylalanine, such as diet drinks. We discussed the basic mechanism for protein synthesis in 168. Your body can synthesize about half of the various amino acids needed to build proteins. There are 10 essential amino acids which must come from the diet. Your body cannot synthesize 8 of them: isoleucine, leucine, lysine, threonine, tryptophan, phenylalanine, valine, and methionine. The other 2, arginine and histidine, can be synthesized, but not in the amounts that growing children need. Other amino acids are called non-essential amino acids because the body can make them on demand. Your body cells can readily synthesize their carbon frameworks. Then an amino group can be attached by transamination or by amination using an ammonium ion as a reactant. Protein deficiency disease develops in people who do not eat adequate amounts of all essential amino acids. All amino acids must be available if the protein synthesis is to take place. Every transfer RNA molecule must appear at the active ribosome in the proper sequence, bearing its individual amino acid. If that doesn't happen, the entire process comes to a halt. A person will be malnourished to some degree if the diet is deficient in amino acids, regardless of its energy content. Examples of protein deficiency diseases include Marsomas and Kwashiorkor. More than 100 million children worldwide have symptoms of these disorders, although none of these conditions is common in the United States today. The figure here and the figure in the next couple modules summarize its metabolic pathways for lipids, carbohydrates, and proteins. The diagrams are present in the reactions in a typical cell. Note, however, that no one cell can carry out all the anabolic and catabolic operations and interconversions required by the body as a whole. All cells differentiate. Each type develops its own set of enzymes. These enzymes determine the cell's metabolic capabilities. In the face of such cellular diversity, homeostasis can be preserved only when the metabolic activities of tissues, organs, and organ systems are coordinated.